In this first activity, we're going to look at how auxin influences root formation, specifically adventitious roots, roots that form from stems or other non-root tissues. This is the same principle that's used in plant propagation when growers apply rooting hormone to cuttings. The key idea here is that hormones don't just work in an on-off way. Their concentration matters, and different amounts can produce very different outcomes. Auxins play a major role in root initiation and early root growth, which is why synthetic auxins like IBA, indole-3-butyric acid, are widely used in horticulture. At low to moderate concentrations, auxin usually stimulates root formation, but at very high concentrations, the same hormone can actually slow growth or harm the tissue. This is a good reminder that plant hormones don't work like simple switches. Instead, their effects are often dose-dependent, meaning the response depends on how much hormone is present. Commercial rooting hormones often come with specific instructions like dip lightly, tap off excess, or use a dilute solution. The goal is to keep the hormone level in the optimal range, enough to stimulate root formation, but not so much that it stresses or harms the cutting. One of the most important ideas in plant development is that the same hormone can produce different outcomes depending on its concentration. Too little may not trigger much of a response, while too much can actually inhibit growth or cause stress because it overwhelms the system and can disrupt normal cellular processes. That's why plants have to regulate hormone levels very carefully. This experiment is designed to show you that balance in a very clear visual way. Before you look at the results of the experiment, I want you to pause and think about this question. Why might a small amount of a hormone produce growth, but a large amount slow it down or even stop it? Think in terms of how cells respond to signals and what might happen if that signal becomes overwhelming or disruptive. Write your prediction in your lab notebook. You'll come back to this idea after you examine the data. In this experiment, identical plant cuttings were used so that the only real difference between groups was the amount of auxin applied. One group received no IBA, another group received IBA at 1,500 milligrams per liter. A third group received IBA at 3,000 milligrams per liter. And a fourth group received IBA at 4,500 milligrams per liter. Everything else, light, water, temperature, and growing conditions, was kept the same. This makes it possible to isolate the effect of hormone concentration on root development. After the cuttings were given time to grow, they were examined for root and shoot development. Parameters such as rooting percentage, root number, and fresh and dry weight for each cutting were recorded and then compared across the 3 treatment groups. This kind of setup allows scientists to directly compare how different hormone concentrations influence root formation in otherwise identical plants. Now you'll use the results to answer these 3 questions in your lab notebook. First, which IBA concentration produced the most root growth? Second, what happened when a large amount of IBA was used? And finally, step back and think about a real-world application. How do the results of this experiment help explain why using too much rooting hormone from a bottle can inhibit root growth instead of improving it? Relate your answer to the idea of hormone concentration and dose-dependent effects. The key takeaway from this activity is that auxin clearly affects root formation, but the amount of hormone matters just as much as its presence. More hormone is not always better. In fact, too much can have the opposite effect. This is a core theme in plant development. Growth and form depend on precise regulation, not just whether a signal is present or not.